Oh, nice. Cool.
All right. So, final review part 2, we are going to go over topics 4 through 7. And obviously, like we're recording, feel free to stop us, ask questions if you have any. And we have a couple of questions for you guys that we'll ask throughout the presentation. Okay. So our first topic, topic 4, is gene expression regulation. This is our triboperon and RNAi pathways. So first thing, transcription factors, you guys probably at this point feel pretty familiar with this, but they are just proteins that will regulate the ability of RNA polymerase to bind to promoter regions on a strand of DNA. So general transcription factors are transcription factors that can just bind to any promoter on a gene. You always need them there. And in prokaryotes, these are sigma factors, and in eukaryotes, these are basal transcription factors. That's just kind of the umbrella names for them. And then we have regulatory transcription factors, and these are specific to certain genes. So like they will only have affinity for certain promoters, and they can be positive or negative, which just means that they'll either increase or decrease transcription when they're bound to those promoters. So you can almost think of like negative regulatory transcription factors as being inhibitors for the promoter that they bind to. All right, so sigma 70 is our main sigma factor in E. coli. It's a general transcription factor, and it binds in 2 places. So I think the diagrams that Laura shows, it kind of looks like a kidney bean where it reaches over on the DNA strand, and one side will bind to the -10 box and the other will bind to the -35. And those are just locations on the promoter. So at one of these boxes, when we go to do transcription, we are going to start unwinding, and our -10 box has more adenine and thymine, and our -35 has more cytosine and guanine. So given that, do you guys have any idea which box unwinding would start at? -10, how come? Exactly, yeah. So when we have our base pairs, adenine and thymine will only form 2 hydrogen bonds with each other, and cytosine and guanine will form 3 hydrogen bonds, which means that it's easier and it takes less energy for the cell to start unwinding at the -10 box. And we're always kind of striving towards energy conservation. And then follow-up question. So sigma 70 is a general transcription factor, which means it can bind to any promoter, but it is also sequence specific. So does anybody have an idea of like how both of those things can be true? Yeah, you're right. Yeah. So sigma 70 is a general transcription factor. It can bind to any promoter. It has a sequence that it prefers or has the best affinity for, but it can still bind any, just with varying degrees of affinity. So if there is one promoter that we need it to bind to a lot, we can assume that it will have pretty good affinity there. And then if there's one that it's not needed at as much, then the affinity won't be as good. So it's just a way of like regulating where it binds the most. And what you were just saying, Brian, was it has a consensus sequence. So a consensus sequence is something that you figure out with like a computational model where just at every position, it takes the base pair that's most common. So, if at, one position you have mostly thymine, and then adenine, whatever, it will just take the most common one wherever, and that's your consensus sequence. So I think on one of our exams, we even had to write one. So just like know what that is. It's kind of a small thing. Yeah. Yeah. Go ahead.
So as long as it has a consensus sequence, counts as sequence.
I mean, yeah, a consensus sequence is just kind of a tool that we use. I guess you could have, yeah.
They're very evolutionarily considered, so like they're not necessarily organism specific. So like for I don't know, some sort of DNA polymerase is really common in whatever bacteria. Like there is a consensus as some sort of like some amino acids goes for that. And for a variety of different reasons that can be beneficial. Like if you're able to like hypothesize like different things because of the presence of the sequences like over and over again. And just overall, it's a very beneficial thing to have. Be it by gene matching, be it by like person specific like binding or whatever. It's just very useful as a tool.
All right, so operons. Operons are something that we find only in prokaryotes, and it's a way that they organize their genomes. And basically what it is you have a group of genes that are related, have some similar function, that are controlled all by one promoter. And when these are transcribed from the DNA, it will make a polycistronic mRNA, which is just a strand of mRNA that will code for multiple proteins when it's translated. And operons are regulated by activators and repressors. And then we see binding of RNA polymerase at the promoter, and repressors will bind at the operator. So on the operon, those are just kind of regions or binding sites. And then operons can be inducible or repressible. So one that is inducible can be induced to function. It's usually off, but you could stimulate it with an activator, or repressible, where it is usually on, but you can turn it off with a repressor. So the TRP operon is repressible. It's usually on, usually running, but you can turn it off with a repressor. So the TRP operon is an operon that codes for a group of structural genes that aid in the biosynthesis of tryptophan. Tryptophan is an amino acid. We need it for protein synthesis.
Why?
What do you mean why? Because we need it for protein synthesis. In this class?
Yeah.
I don't know.
Well, okay, so the bacteria we look at in theory for like
Oh.
that specific for E. coli.
Yeah.
Yeah, for E. coli specifically for this class, I guess not specifically for this class of E. coli, but they need tryptophan in order to survive. It's like an essential amino acid for their protein function. And so for this class specific though, like it's very important that you guys understand the level that you'll get to progression. Sometimes you forget that that's like an essential thing for the tripopon specifically. It's like kind of a big concept.
Yeah, it's fine. All right. So just briefly before we get into the details, the TRP operon will behave differently depending on our levels of tryptophan in the cell. So if we have a ton of tryptophan already available, then tryptophan, which is our repressor for this operon, will bind at the operator, and it will prevent RNA polymerase from binding, either by like obscuring part of that binding site, blocking it, or it will make it, I don't have any board space, but it will make it essentially like a roadblock. So our RNA polymerase can't pass that repressor where it's bound. And then we won't have transcription because we don't need those genes to make more tryptophan because we already have it in the cell. We don't want to waste energy making something that we already have. If we have really low tryptophan levels, then RNA polymerase will bind and we will have our mRNA transcribed and translated, and then we'll be able to make more tryptophan. And then at medium levels, things get kind of weird. RNA polymerase will bind because we probably won't have enough for tryptophan to be bound on that operator to repress it. But then it will cause attenuation during the process, which just means that like transcription is going to end early before the mRNA strand is complete. So we'll have like a partial strand, but it won't finish all the way. So this is the structure of the TRP operon. What I was saying earlier is that this is our operator and this is our promoter. And our RNA polymerase moves this way towards transcribing our structural genes. So when we have TRP bound on our operator, acting as our repressor, our RNA polymerase will be bound here, and either tryptophan might bind right here and kind of get in the way of RNA polymerase binding, or it'll just make it so RNA polymerase can't be passed. So that is how it acts as a repressor. Go ahead. It's the amino acid. So tryptophan is an amino acid and we use it to make proteins. Do you feel okay with that? Because if you have another, okay. All right, so our trip operon also has the ability to make structures called hairpins or loops, and that is because regions of our mRNA strand will have base complementarity, so they can kind of fold up, and based on where these hairpins form, it will have different effects that may cause attenuation. If we have medium levels of tryptophan and we decide halfway through transcription that we actually don't want to make our full-length mRNA. So to go into a little more detail, if we have low levels of tryptophan, then our RNA polymerase will bind our promoter, it will move down the DNA, and because we're talking about prokaryotes, this isn't happening in the nucleus. We don't have a nucleus, which means that our RNA polymerase and our DNA and our ribosomes are all in the same place. They're just floating in the cytosol in the nucleoid region. And so our transcription and translation will happen simultaneously. Our RNA polymerase will move down the DNA strand, start making our mRNA, and then as that mRNA is being made, a ribosome will bind and start translating it as further down the DNA strand, it's still being transcribed. So we have our ribosome following our RNA polymerase. Our RNA polymerase will transcribe the 1 and 2 regions of our hairpin, and then because they are complementary to each other, they will form our first little loop. And because when this is folded, our RNA polymerase can't really move down the strand anymore, it will pause right before that first region where our hairpin is forming. And then RNA polymerase will keep moving, it will form region 3, and then we'll have region 2 and 3 together. So our 1-2 hairpin will break, our 2-3 hairpin will form instead, and our ribosome will move a little bit further. So now we have a 2-3 hairpin, and our ribosome will sit on the trip-trip codon of our region 1. And what that is a place that holds 2 trip tRNAs. So as the ribosome is transcribing this protein, it needs our trip tRNA to give it tryptophan. But if we have low levels, then there won't be trip tRNA right away available to give that to the ribosome. So we'll kind of pause at this codon while the RNA polymerase keeps moving. And once we have that 2-3 hairpin form, that just essentially tells the operon that, like, okay, we don't have enough tryptophan, so we're going to continue. It's an anti-termination signal. So we end up making our full mRNA, and then eventually when those trip tRNAs come along, the ribosome will be able to move on from region 1 and translate the whole set of structural genes. So here is an image of that happening. Our RNA polymerase will make regions 1 and 2. They will snap together in a hairpin. Our ribosome will get stuck right before that. And then, because here, these are our trip codons, we don't have tryptophan to give the ribosome to make our protein. It will stay here for a little while. Our RNA polymerase will make our region 3. Our 2-3 hairpin will form. Eventually, our 4th region will form. And when we have trip tRNA eventually that comes into here, our ribosome will keep moving and we'll end up with a full strand. Yeah. We okay with that? All right. So at medium levels of tryptophan, it's going to start off pretty much the same. We won't have anything bound to the repressor. Our ribosome is going to move, stop before our 1-2 hairpin. And then we have usually enough at this point that there's trip tRNA available, so it won't pause at region 1. It will move past those codons because at those codons, trip tRNA is able to give tryptophan to the ribosome, so it can keep moving, keep synthesizing our protein, and then it will sit on region 2. And when it's sitting on region 2, that means that region 2 can't fold into a hairpin. So as RNA polymerase moves along and we form our 3rd and 4th regions, we'll end up with a 3-4 hairpin instead. When we have our 3-4 hairpin, that's signal to attenuate. And essentially just what happens is when that folds, our 3rd and our 4th regions, it will snap our mRNA out of the active site of RNA polymerase. So we end up with only a partial strand. Here is that happening. Yeah. So we have our hairpin form initially, then we pass really easily by our trip codon. So we sit on region 2 instead. We can't have our 2-3 hairpin because 2 is occupied. So instead we have a 3-4 hairpin, and just the way that that folds, it snaps the mRNA out of RNA polymerase.
Wait, you can choose to stop codon and then you can what?
Region, oh yeah, so region 1 is our trip codon. So this is where we need trip tRNA to give to the ribosome. And then region 2, there's just a little stop codon where we'll pause if we make it past our trip codons. Go ahead.
So does the 2-3 hairpin at this point?
So here, let's see. So when we have our ribosomes stop at the trip codon because there isn't trip tRNA, then region 1 won't be able to form a hairpin because it has the ribosome sitting on top of it. So then the 2-3 hairpin will form because our region 2 will be made, our region 3 will be made, and they'll be available to go together. But in this case, we don't have that hairpin form because we have our tryptophan paused on our stop codon. So then we end up with 3-4. Okay, is everybody okay with that? These are kind of like, I would draw them. Okay, so those are the basics of that. I didn't put another slide for high tryptophan because then it would just be repressed and we wouldn't have to worry about any of the hairpins or attenuation.
Can I ask a question about that?
Yeah.
Okay. If the trip levels are high, like, it is a concept that the repression mode or tryptophan will bind to repress. Can someone explain to me why/how? Like, how does the tryptophan bind to the repress? Yeah, high levels of tryptophan will repress the compound, but how? Yeah, I want to know more about that repression. Okay, so what happens is, Laura likes talking about this earlier on in class, so, I mean, it's been a couple of topics, so it might not be fresh. But it's something that stuck with me since, like, I took the class back when. This concept of like us seeing a drawing and thinking, oh, obviously it looks like the tryptophan is just gonna be added there or whatever. These molecules don't have eyes, right? So they don't know like where they're going. They're just like flowing around in the plasma, right? They're just like there. High concentrations of tryptophan allow for you to have a higher chance of the tryptophan running into this repressor at random. It is by random chance that it happens. It's not like something goes in there and it's like, oh, I can find the tryptophan here. I'm gonna like grab it. It doesn't grab it. It's just like the more you have of it, the easier it is for, let's say, like Laura really likes doing this. She like walks around and she'll be like, okay, so the more tryptophan level there is, the easier it is for this to happen. Okay. This one. But basically what I'm trying to say is the higher the levels of the molecule, the easier it is for it to like randomly bind to the
Also something we saw in a problem set last semester that she gave is like, I guess everything in the cell, just because it has kinetic energy and thermal energy from whatever temperature the organism is operating at, we always have random motion. So things are just happening by random diffusion and it may or may not encounter that repressor. And then obviously at higher concentrations, the probability of that increases. The word for that, just like random motion in the cell, is Brownian motion. I don't think that you'll need to know that, but that has come up before and people have had questions about it. So okay, yeah, non-coding RNA. So this is just like any RNA that doesn't code for a protein. It's in the name. They aren't translated. They have other regulatory roles. And this was an exam question last semester that a lot of people weren't sure on. Basically, a non-coding RNA is anything other than mRNA, and a coding RNA is mRNA. mRNA codes for a protein. It's translated into a protein. So like that's all that is. We have a whole bunch of examples. I would know a few of them, just like, have some written down or memorize them. So rRNA is ribosomal RNA, and it's the catalytic part of a ribosome. So a ribosome has a protein component and an RNA component, and this is like really new. We didn't used to know that this could happen, but the ribosomal RNA is the catalytic part. It's not the protein that catalyzes the reactions happening in a ribosome to make the protein. So that is called a ribozyme. That is a term that you should know. And a ribozyme is just any RNA with enzymatic function. So RNA that can catalyze a reaction. tRNA is what we just talked about. It will bring an amino acid to the ribosome during translation to help make protein. Small nuclear RNA, it's part of the spliceosome. It's also a ribozyme. I have a couple of other examples here. We're going to send out these slides. So just like take a look at them, know a few of them. You don't have to know them in detail, just be kind of familiar with a few. So non-coding RNA is what we use in RNAi, RNA interference. It is found, or it's a process that's found in eukaryotes, and it is a post-transcriptional method to regulate mRNA. Usually it's used for suppressing genes, and the reason that that's relevant and useful for us is because in research, we can use RNAi to knock out genes. So like if we were doing diabetes research, we could use RNAi to knock out like the leptin gene in mice, and that would just make them keep eating always, and then that would induce eventually diabetes, and we could study that. So there are just lots of research applications for RNAi. And also, like when we want to know what a gene does, the easiest way to tell what a gene does is by breaking it and to see what isn't happening anymore. So this, I like could see there being a word problem or a short answer question on, like, that is that application research. That's why we really care about RNAi. So RNAi uses RISC. It's the RNA-induced silencing complex, and it's found in both of the pathways that we're going to talk about. And what RISC does is it targets a specific strand of mRNA that we want to silence, and it prevents it from being translated to a protein. So sometimes it will do that by sequestering it, sometimes just inhibiting it, sometimes by degrading it altogether. But RISC is going to bind with an RNA guide strand, and that RNA guide strand will have complementarity with our target strand. So when our guide and our target bind, RISC is going to silence somehow the target. And Argonaut is a part of RISC. It's a subunit of RISC that is the catalytic portion. So that is especially important because it's going to be like cleaving the mRNA that we want to silence if it's being degraded. So miRNA, microRNA, is endogenously expressed, which means in the organisms where we find it, it is encoded in their own genomes. And it's a guide strand for RISC, so they will form a complex together to target mRNA that we want to silence. And it's really short. It's like 20, 25 bases worth of double-stranded RNA. Just gonna take a drink. Halfway. Sorry, I tried to fit all this on one slide, but it's a little cramped. So in our miRNA pathway, we are going to start in the nucleus because this is encoded within the genome of the cell. And RNA polymerase II will transcribe a pri-miRNA, not pri, and that's our initial transcript. Then DROSHA, which is an RNA nuclease, will process our pri-RNA into a pre-miRNA. So our pre-miRNA can be exported into the cytosol using exportin, which that is unit 3 stuff, which I think they probably went over last night that you should also be familiar with for the exam. And once we're in the cytosol, we will have dicer, which is another RNA nuclease that will cut up and process our pre-RNA into our 20 to 25 base pair duplexes. And the duplex is just the name for like the short strands that are double-stranded, base paired with each other. And our duplexes will bind RISC. RISC is going to unwind our duplexes, and then one of them will be our guide, and our other is just going to be degraded, usually by Argonaut. And so then our guide strand will search for targets, and if it has complementarity, our guide and our target will bind through just base complementarity. And usually for miRNA, the match won't be exact. It will just be close enough and it will have enough affinity that they will bind. And that will either suppress translation or sequester that target strand into the P-body. So it can't be translated. It's separated from the ribosome. The P-body, you don't have to know a lot about. I don't know if Floristil calls it this, but it's kind of like a broom closet. We don't really know what happens there. It's just where we put stuff that we don't have any immediate need for. And in the event that we do have an exact match, that is a sign that that strand should be degraded. So that is when Argonaut, our catalytic subunit, will cleave it, and that mRNA will be degraded. Our target mRNA will be degraded. siRNA is very similar. This one is exogenously expressed, so it is not included in the cell's genome. Usually it will come from a virus. And a lot of times the use for this is like protecting against other DNA-based viruses, because it will cleave that target mRNA. And same thing, it's a guide strand for risk and 20 to 25 base pairs. So this time we're skipping our first step. We're skipping our DROSHA processing step. We're skipping our pRNA step because we don't have any of that transcription happening in the nucleus. We're just coming straight into the cytosol because it's coming from presumably a virus. So then it will be processed by dicer into duplexes, and those are going to be the same length. RISC is going to unwind it. We're going to pick our guide strand. Our guide strand is going to find our targets. And usually with siRNA, we're going to have exact matches, which means that we will have degradation by Argonaut of our target strand. Do you feel okay with that? Okay. So here's just a diagram comparing the two. From here, they're basically the same. The only thing that we have different is for miRNA, we're starting off in the nucleus with our genome, and we're transcribing it into RNA, and then we're exporting it into the cytosol, and from there, they're pretty much the same. Okay, that is topic 4. Do you guys have any questions on that?
Argonaut always.
Argonaut is a part of RISC, so yeah. It's just a subunit on RISC, and it's the catalytic subunit. Everybody else good? All right, John is going to take over.
All right, how are we feeling? Tired? That sucks. Welcome. All right. This specifically is my favorite topic. You don't have to like it. You can hate it if you want. I don't care, but it is very important. So we are going to be talking about the 4 different pathways that we went over. Start with RTK, MAPK, cell survival signaling, JAK-STAT, finish with GPCR. The slides will be sent out if you just want a general broad, like bird's eye view of what each does. There's that, but we're about to go into a little bit more detail. So RTK, RAS, MAPK pathway, or the growth factor signaling pathway. It's one of the more broader ones that we have. So first off, what RTKs are. So they're monomer receptors. That's like the full definition for it. Monomer receptors with an extracellular receptor binding domain and intracellular tyrosine kinase domain. What does that mean? It has a receptor outside the cell, and it has kinase activity at the bottom. So like that. And how RTKs work. You need a signal to bind on the extracellular portion for then the 2 molecules to dimerize, which is to form like a single complex. And once that happens, they are able to create binding sites for intracellular signaling proteins, and then farther stuff happens downstream. So mechanism of RAS signaling. So once the signal binds, and then the RTKs get active, they phosphorylate each other, and then the protein Grab2, or the adapter protein, however you want to call it, goes in and binds, and then has an SH3 domain, which is how the SOS protein is able to bind. People call it sauce. I hate that name. So SOS just sounds better. And then SOS goes in and it's able to activate RAS, which then is able to get its GTP in instead of GDP. And then transition the signal happens. Very important here. What's an SH2 domain? Come on, I know one of you knows it. Come on. Anybody? It binds something specifically. That's SH3. Anybody? Okay, so what's an RTK? Receptor tyrosine kinase, right? An SH2 domain is a phosphotyrosine binding domain. So it has to bind a phosphorylated tyrosine. Very important. It's something that just like, you guys don't remember it probably because it's so long ago, but it is very important for you to know that because the GRB2 protein is only able to do its job of like relaying the binding if that tyrosine is phosphorylated. Also, what do we need in order for phosphorylation to occur other than a kinase? Not just in general. In general. Like what's something that's essential for you to have phosphorylation occurring? You need a hydroxyl group. So there have been questions in the past that have asked students, at least when I took the class and I got the question wrong, which is why I remember it. Laura might ask you like, oh, out of all of these, where would phosphorylation happen? And you need that oxide group or the hydroxyl group present. Tyrosine is a good example of that. If you don't remember what tyrosine looks like, it's like a phenyl group with an OH extending away from it. And you need that hydroxyl group in order for your inorganic phosphate group to bind to your molecule. And so back to track, once your tyrosine is phosphorylated, then the SH2 domain binds. And then the function of that GRB2 protein is literally to relay this and then get this, which is the PXXP domain, to bind right here. And so basically what's happening here is you are forming a SH3 PXXP motif. And PXXP is just proline, whatever, like that literally means proline, whatever amino acid, proline. It's like a proline rich like region, and you're able to bind the SH3 domain. Yes? It has both. It has SH2 to bind to the phosphotyrosine, and it has SH3. Yes? For the tyrosine? Well, it's just like the outermost part of the Lily's gonna draw it. It's the last amino acid or the last group that extends away from the amino acid. That's a really good hexane. Yeah, and so oh, go ahead. Yeah. The phosphotyrosine. Yes. SH2. Yes. I think. Don't quote me on it. We talk about SH2s more. But proline rich, anybody know what a proline looks like? Anybody, you don't have to draw it. Anybody know a cool fact about proline? Yeah. They're helix breakers. Yeah, because they form. Okay. Okay.
Yeah, well, it's good. So proline specifically, a molecule, because and it cycles around. That's and it just, forms a covalent bond with the backbone atom, which is how the secondary structure is. And so when like the molecule is like this, and it's here a little curve, and it comes back up to you guys, so there's really there, so very close to
In RTX, you know that's not to be obligated seeds, it's a different kind of secondary structure we talked about. It's a polyproline elements, so those are the elements. And proline, I got people like, you don't know that, but that is. That are.
Any questions? I'm sorry if I moved a little everywhere. Okay, and so grab 2 is recruiting RAS Gef. Yesterday I went over what a Gef is. Anybody feeling brave? Yeah. Yep. All right. And so it's gonna go in there into the RAS that's anchored to the membrane. And so once that RAS becomes active with GTP in its domain, then, or in its binding domain, then signal relay happens. And so after that comes the fun part, I guess, of this signaling pathway. So active RAS is going to activate MAPK3. Can someone tell me how? We don't know. Really good. And so somehow, some way, because it is not a kinase, activates MAPK3. And that then leads from MAPK3 to activating MAPK2. And once that's active, it activates MAPK1. And then it's able to relay signals to whatever different activities the cell needs. In our class specifically, we talked about changes in protein activity and changes in gene expression. But MAPK1 can also phosphorylate other stuff. All right. Cell survival signaling, my favorite pathway. All right, so what's it do? So the default for our cells, kind of sad, they're supposed to die. And for the cells to remain alive, you need a signal to be constantly sent in order for them not to go into apoptosis. And so this pathway specifically works really well with other pathways in order for cells to survive. It is not the only thing that needs to occur. So how does it start? You'll get the survival signaling, you'll get the survival signaling molecule to bind to a receptor. And these receptors are RTKs, and so they will cross phosphorylate. And then once they are both active post cross phosphorylation, an activated PI3 kinase will bind. And that is able to activate the inositophospholipid molecule that will be lodged into the membrane over there. And then it will get phosphorylated, becoming PIP3, which is just 3 phosphates. And so it is, and PIP3 and PIP2, they're not proteins, they're lipids. Very important. And so continuing, for this second stage to happen, you'll need another PIP3 in order for PK1 to bind to. And also for the ATK to bind to. So you need 2 of them to exist beforehand in order for this to occur. PK1, once lodged to PIP3, will then activate one side of ATK, or AKT, sorry, AKT. And once that happens, then there's a conformational change that happens on the AKT, which then allows for it to be phosphorylated by the second protein kinase, in this case, mTOR. And that's that over there. And then, once that happens, once the AKT is fully activated, it then dissociates from PIP3. So that's the green molecule over there. And so the activated AKT will then bind to BAD, which will phosphorylate it. And counterintuitively, it will inactivate BAD. So BAD's main function is to sequester BCL2, which is a protein that inhibits apoptosis or has further functions downstream that lead to an activation of apoptosis. And normally when BAD is not inactive due to phosphorylation, it is sequestering the protein. What does that do? It's just holding onto it. It's not allowing BCL2 to go do whatever else it needs to do. Once BAD is phosphorylated, it then binds the 1433 protein while phosphorylated, and then the BCL2 then becomes activated, enabling it to do whatever it is that it does in order for apoptosis to not occur. We are not going to dive deep into these concepts. They are harder concepts. If you want to look into it, you can. No.
So if you want to, or if you receive. So you want to stop this happening, don't want to give a cell death, you want the cell death. And you want that to happen after 50 cells. How can you then continue after? How, so you can get rid of the phosphate, right? If you want to go back to an active, how do you do that?
What enzyme?
Called P10 and you should write it down and have it on your notes, you can know it, just in case, that's one of the things you might throw on, like if you don't know it. So P10, P10 is the phosphatase that takes the last phosphate off of everything. It dephosphorylates and goes back.
All right. And so this is just another slide on BAT and BCL2. Talked about BAT and BCL2 function. So there are two possible outcomes. This is just like an easier way of like getting the final step into the note card. Active BAT inhibits BCL2, which then leads to cell death, or inactive BAT will not be able to sequester BCL2, which then will lead to cell survival. All right, and that's just an overview of the pathway without it being all chewed up. JAK-STAT, okay. Oh. All right, so involved in many processes. For this class, we will be talking about transcription of milk genes. And so most important part out of this entire pathway, JAK receptors aren't RTKs. The whole reason why JAK exists is because its receptor doesn't have a kinase domain. And so it can't cross/autophosphorylate, and it can't relay signal information because it is not able to phosphorylate. And so once the prolactin receptor in this case binds, the two receptors dimerize. This dimerization causes the activation of JAK. Does it say over there? No, okay. What's JAK stand for? Raise your hand, Matron. Apologize to Lisa. There you go. Okay. So yeah, just another kinase. They initially thought it was just like irrelevant, and then they found out its activity. And so once cross-phosphorylation occurs, then the JAKs will then phosphorylate their own receptor molecules. And then once that happens, the active STAT will then be able to bind onto the phosphorylated receptor through its SH2 domain, which is the phosphotyrosine binding domain. And once they are bound, then the JAK will phosphorylate that molecule, so the STATs. And then once active, that will cause a conformational change, which will cause them to diffuse away from the receptor, and then they will be able to bind each other. And so once the dimerization of STAT happens, then it goes into the nucleus and acts as a transcription factor in order to upregulate the production of the milk protein. So this pathway is pretty straightforward. It's more about, and JAK function and that the receptor is in RTK. Any questions? No, all right. Oh. Yeah, I mean, I basically just talked for all that. Okay, and then finally GPCR pathway. It's one of the most common, or it is the most prevalent one in like humans. And whatever data Laura gave about new drugs being processed and made, most of them target GPCRs. So they have 7 transmembrane domains. It's that squiggly structure. And they are bound to a G protein heterotrimer, so alpha, beta, gamma. But most important one is probably G alpha. G alpha is a GTPase. And what does a GTPase do? Yeah. 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 And what's that lead to? Okay, there you go. So in its inactive state, G alpha is bound to GDP. And so this is just like a large walkthrough of the pathway first. Ligand is going to bind to GPCR, which then leads G alpha to diffuse away from GDP. And so the GTP is then able to diffuse into the G alpha, activating it. And so once active, the G alpha is going to dissociate from G beta, G gamma, and that remains as a dimer, so beta gamma dimer. And so that complex will then bind effectors to start a signal transduction pathway. So that's what happens down here. Down here, sorry. Yeah. And so basically at the end of the day, once the signaling needs to cease, deactivation of G alpha is going to occur when RGS is going to bind to it. And so this increases G alpha's GTPase activity, making hydrolysis faster. And so its GTP bound state will then become a GDP bound state post hydrolysis. And so once inactive, G alpha is able to diffuse back towards the gamma beta complex and reform the heterotrimer. So GPCR is the GEF analog and RGS is the GAP analog for this specific signaling pathway. I would write that down just in case you don't want to like waste your time looking and reading through all the entire topic notes. It's just an easier way of just, oh, this happens here. Let me go right to it. So that's a closer up, newer image of like a drawing of the pathway. And the squiggly lines look a little bit more three-dimensional on that drawing. But overall, it's just the same cycle. The important part is that G alpha, once active, will dissociate away, and then signaling can be done. Some common misconceptions. This is broad. This isn't specifically about GPCR. So GAP and GEF, neither of them are enzymes. GEF promotes GDP dissociation and then allows GTP to diffuse in. It doesn't dissociate GDP. And so the GAP binds to GTPase and activates its intrinsic catalytic ability. And then hydrolysis occurs, but GAP itself doesn't hydrolyze GTP. And so these signaling pathways, they're all very, very much controlled, and signal transduction is also very well controlled because you do not want a defective signal to be amplified and cause more damage. So yeah. All right. Moving along. Nice. Break your screen. Moving along.
Okay. So this is topic 6, cytoskeleton, microtubules, maps, all of that fun stuff. I like this topic. So just briefly, there are 3 types of cytoskeleton. Our first is actin. If you've taken like kin or physiology, then you're probably pretty familiar with it. It's really involved in muscle contraction. With myosin, it has to do with cell motility, cytokines, maintenance of shape, just like as a structural feature. Intermediate filaments we really don't talk about, but they are also for just shape and structure. And then microtubules are the ones that we're going to talk about the most. They are really rigid structures, so they can resist compression on the cell. They are our spindles that will pull our replicated chromosomes or our sister chromatids apart towards spindle poles. And we will talk a lot more about them now. So our spindle here, there are 3 types of microtubules that make up our spindle during M phase. The first one is our aster microtubules, and they grow out from our centrosome right here in all directions and attach to the cell cortex. So the way that I remember this one is an aster is a flower that looks kind of like a daisy. So like the petals fan out in all directions, and that's kind of what the aster microtubules look like. The kinetochore microtubules just kind of point inwards from the centrosome, and they are the ones that are attaching to the kinetochores of sister chromatids in the middle when we line up during M phase on that like imaginary hemisphere line. And then our interpolar microtubules are overlapping at the spindle, and they will have some motor proteins attached here, and they are the ones that like grow and shrink and are kind of pushing and pulling our cell apart, especially when it's time for like cytokinesis. Okay, so microtubule structure. Microtubules are made of tubulin heterodimers. So there are alpha and beta tubulin subunits, and they are polar molecules with a plus end and a minus end. So when we have our microtubule, right, we have our alpha, or not our alpha and our beta, we have our plus and our minus end. We can have tubulin addition at either end technically, but the reason that they're called plus and minus isn't because they're charged, like positive and negative charge. It's because at our plus end, our tubulin addition is going to be really rapid, and at our minus end, it can still happen, but it would be really slow. So like in the example of our kinetochore microtubules, our minus end is going to be embedded in the centrosome, and then our plus end is going to be reaching out because we can polymerize really quickly to grab towards our kinetochores. So when we put together tubulin subunits to make a big long strand of them, that is called a protofilament. It's kind of just like a string of tubulin subunits. And we take 13 protofilaments and we put them together in a lateral circle to make our actual microtubule. It's hollow in the middle. It's kind of cool. You can see like on an electron micrograph, looks like a straw. And our microtubules will extend out from our microtubule organizing center or complex. Yeah, so this is kind of what I just mentioned, even though it's possible. And like in vitro, we would see a little bit of growth from our minus ends. We won't in vivo in a cell in something alive because it's going to be embedded in our MTOC. But normally, and this is really important to know, and this is something that people kind of get confused on a lot on exams, for our class, and especially for our polymerization assays and graphs, we are not talking about an actual cell. We're talking about a test tube. So unless it says otherwise, there's no MTOC, it's just tubulin subunits and GTP in a test tube. All right. So more just about the structure of tubulin. Tubulin is a GTPase. It is an enzyme that can hydrolyze GTP to GDP. Both of our subunits have binding pockets for GTP, but you can see on that binding interface between them, hopefully, that our alpha tubulin has a GTP binding site, but it's kind of buried. So once our alpha and beta tubulin come together, that GTP is just going to stay there. We can't really hydrolyze it just because we don't have access to the site. And once these two monomers form our heterodimer, they're just not going to come unbound. They're covalent and they have really strong affinity. So this hangs out. And then our active end, our plus end, is our beta subunit that has GTPase activity still because it's not obstructed by that interface. Okay, so more about our microtubule dynamics. Again, this is in vitro, in a test tube, not in vivo in a cell. We make these graphs by taking a test tube and some substrate and then adding purified tubulin and GTP. And then what we do is we use a spectrophotometer and we shine light through the test tube at different time intervals. And as the amount of microtubules in our test tube increases, our spectrophotometer will measure that the absorbance of light that it shines through increases because there are more microtubules present to absorb that light. So that's how we get those values. And then we make a graph of absorbance versus time. So the more microtubules, the more absorbance. And we have 3 phases during our little assay. Our first is lag, our second is elongation, and our third is plateau. So of those 3 phases, which one do you think would look the most different in vivo in a cell? Which one? How come? Exactly. So this was something that like people had a really hard time with on our last exam. Lag phase is caused, and I'm going to get more into this, basically by starting from scratch, de novo. We have no MTOC, and we have to take our tubulin for the very first time and form our protofilaments, so form our longitudinal bonds that extend it, outwards, and also, coordinate our protofilaments so they form those lateral bonds to make that straw shape. And that takes a lot of time because we're starting from scratch. These all have to find each other, especially before, we hydrolyze our GTP. And in a cell, we'll have like a centrosome and we just won't need to do that. So in a cell or with an MTOC or with a microtubule seed, anytime we're not starting completely from scratch, we will not have lag phase. That is really important to know. Write it down if you're not like 100% sure of it. If you have questions, ask them. If we don't have an MTOC, then we will have a lag phase. But if we have anything to build off of, lag phase will not happen. All right. So yeah. In a test tube or anytime that we don't have our MTOC or our seeds, lag is just, the beginning of that formation. It's forming those initial bonds. And the higher amount of free tubulin that we have, the shorter our lag phase will be. So that's kind of like what we talked about with tryptophan earlier. The more free tubulin we have, just because of that random motion or Brownian motion, whatever, the higher the probability is that they're going to bump into each other and bind. So higher concentration of free tubulin, it means lag phase will be shorter because all of these interactions can happen more readily. Once we have our seed formed and we don't have to worry about making all of those really complex interactions and like forming that initial geometry, we'll have our elongation phase. And that is when mostly our plus end is going to start really, really rapidly adding GTP tubulin. And as our microtubules elongate, we're going to be using up our free tubulin. So eventually that rate of addition is going to slow down because there just won't be anything to add on anymore. So after elongation, we will have plateau, and that is like when we've used up the majority of our free tubulin. We're not actively growing, but we're not depolymerizing either. Our rate of addition is just completely equal to our rate of depolymerization. So this is what we call dynamic instability. They're not just like hanging out, not adding and subtracting. We're just in equilibrium between addition and what's the word? Depolymerization. Go ahead.
So in the cell, there's always going to be
Well, I mean, our cell, like we have our centrosome, that is our MTOC. It's just any nucleating site that our microtubules will build off of. So in a test tube, we will not have lag unless we add a nucleating site, but in our cell, we will have it.
So,
Gamma tubulin ring complex, or our like whatever we're building off of in the cell, it will be there, but it's not guaranteed in vitro. Okay. So let's see, do we? No, we don't. Okay. So what happens in vitro, say like 5 hours into plateau phase? Right, yeah. So the word for it is catastrophe. Catastrophe is when we have complete depolymerization of a microtubule. The reason that that happens is because tubulin is a GTPase. So for our microtubule to be stable, the very end of the microtubule has to have a cap where tubulin is still bound to GTP. And as our microtubule grows, the sections that have been put on first will eventually start to hydrolyze their GTP to GDP. And if that catches up because we've stopped adding or our rate of addition is too slow, whatever, eventually we will only have GDP and we will completely depolymerize. It will be catastrophe because in a test tube, we can't replenish our GTP. In the cell, if we want to keep that microtubule built, then we'll have GTP available so we can keep that cap maintained, but there's no enzymatic mechanism in a test tube once you've hydrolyzed all of your GTP to get more of it. So would we normally see catastrophe in cellular conditions? You guys want to argue it out? You don't have to.
No, explain why. Which she has to go on. Does catastrophe happen now? Well, I mean, that's
Depolymerization does, but normally not catastrophe. So depolymerization is just any shrinking. And say we lose on our microtubule our GTP cap, say that we want, if we have our kinetochore microtubule that's trying to find our kinetochore and our sister chromatid, it might reach out too far this way because it doesn't have eyes, it doesn't know where it's going. So it needs to depolymerize. The dynamic instability is a good thing, but then since we have more GTP available, it can start to polymerize again, put that GTP cap back on and find our kinetochore. But in a test tube, we, again, once we've hydrolyzed all of our GDP, don't have any way to get it back, so we'll have catastrophe. Make sense? All right, so I've talked a little bit about this already, but dynamic instability is just the idea that microtubules can grow and shrink independently of each other. Just because it's growing now doesn't mean that it always will be. Just because it's shrinking now doesn't mean it'll be a total catastrophe. We can go back and forth and kind of reach out and in, and that's really important, especially during mitosis, because when we're forming those attachments to our kinetochores, we need to kind of reach out and then pull back and reach out and pull back until we form our correct bonds. So whenever we have more GTP hydrolysis than tubulin addition, that means that we're going to have depolymerization because we don't have a cap anymore. And whenever we have more tubulin addition than GTP hydrolysis, that means that we'll keep that fresh cap of GTP tubulin, so we'll be in a state of growth. And then these drugs we don't talk a lot about. Some of them will stabilize microtubules, some of them will destabilize them. I'm not going to go super in depth. I would maybe write down what they do, especially Taxol and maybe colchicine. I think that those are the ones that Laura will use the most often. But yeah, mostly Taxol. Vinblastine we don't really talk about that much, but let's use Taxol for an example. So Taxol is a chemotherapy drug. If we give or expose a cell to Taxol, it will bind and stabilize microtubules, and that means that they can't depolymerize anymore. So how could that be used as a chemotherapeutic agent? How could stabilized microtubules prevent or treat cancer? Go ahead.
Those windows.
Well, they can add, they just can't depolymerize. So you're on the right track. We can start to form spindles, right? But if they don't form the correct attachments to our kinetochores, they can't retract backwards and then add back on and head in the right direction, which basically just means that we're not going to be able to form our proper attachments because we don't have that dynamic instability that we need to reach out until we bind at the right place.
Well, they have a shot. They're extremely
Well, yeah, but like especially not for all of them to do it at all of our kinetochores for our sister chromatids. And so what happens is if we get stuck for too long in M phase, the cell figures like, okay, something is wrong here, we're not dividing right, and then it will undergo cell death because it doesn't want to risk during division dividing with some sort of damage. So if we have a cancer, which is an overgrowth of cells, by stabilizing our microtubules, we're halting it in the middle of its division cycle. And if it stays there for too long without building its correct attachments, then it will die. So that is the kind of question that I would be prepared for with these. Okay, yeah, maps, maps and motors. This wasn't my photo, but I like it. Okay. MAPS are microtubule associated proteins. I think the best example is tau. It is expressed in neurons in the central nervous system. If you've ever learned about like Alzheimer's or any neurodegenerative diseases, you've probably heard of it. And it is used to stabilize microtubules. You don't need to know a ton about that. I think the only reason that we bring it up is because it's pretty relevant in like medicine and research, especially with neurodegeneration.
Well, we need to know, that once tau, if there's a misfold or if tau associates, they agglomerate, and then that leads to, like misfold protein being repaired.
Yeah, so
I'm gonna talk.
I mean, how far into it? I can explain it, but I don't know how far in depth we'll
Oh,
have to
I'm
get.
just wondering.
I don't think I have a slide on it.
Basically, I mean, Lily is way more clear with it than I am, but, so you just need to know that tau stabilizes the microtubule by binding to the microtubule. And so once this stability is removed, because the tau binds or because it's old or whatever, then, tau agglomerates into,
They call them tangles,
Yeah.
aggregates.
And so it kind of looks, if you look at like a microscopy image of it, they look like seeds. Like in group. They're like darker spots within like the middle looking around cells. I don't think we have pictures of them.
No, I mean, I don't think that we really have to go that in depth into it, but basically, tau is an intrinsically disordered protein, which means that it doesn't have tertiary structure. And these types of proteins are really not at all prone to misfolding just because of their chemistry, but sometimes they will, especially in neurodegenerative diseases. And when they do, they can form protein aggregates like we saw in like units 1 and 2. And when they aggregate, because they occur in like the central nervous system, that will induce other tau to aggregate and they form this big clump. It's a fibril, a tau tangle, and it basically gums up the cell until it cements the entire inside of it with misfolded, tangled up tau, and then it kills nerve cells, which is like how you end up with things like Alzheimer's, or it contributes to, is associated with Alzheimer's. Yep. It's cool. It's fun. Okay, yeah. So motors. People have a really hard time with motors. I think they're really cool, but there's something that like it might take a little practice to visualize. So the analogy that Laura uses is that motors are like cars on the highway that is the microtubule, and they can be used to transport things. They can be used to attach to 2 microtubules and pull them relative to each other. And they all have directionality. So some of them will walk towards the plus end of the microtubule and some of them will walk towards the minus end. Our kinesins, they are the ones that look like they have feet. I think Laura showed a video of that in class. They are plus end directed, except for kinesin 14. That one moves towards the minus end. And I think maybe kinesin 5 doesn't have directionality. We'll see that in a second. Dyneins are the opposite. They walk towards the minus end and they're really important in motility. So like cilia and flagella, they'll be the ones causing that movement. Okay, yeah. So here is, oh no, that's what I was thinking, kinesin 5s. Here's like the general rundown on kinesins. I would write these down maybe too, like the non-coding RNAs, just to kind of be familiar with what they do. Our first couple are kinesins 1 and 3. They're different families of the kinesin protein. They're mostly involved in cargo transport. our kinesin 5, that's like egg 5, and they will walk on microtubules at one end and then grab them on the other, so they're tetramers. It's kind of like they have feet at both ends. And our kinesin 13s do not walk processively on the microtubules. Those are the ones that I was thinking of. They just kind of diffuse along and destabilize the plus end. And they're used in anaphase A to shorten our kinetochore microtubules at the plus end to kind of pull them towards the spindle. And MCAC is an example of that. So if you don't remember that, I think they're going to go over that Sunday. And then just here's a diagram of that. I drew this on my note sheet. I think these are fun. But again, other than egg 5, you don't need to know a ton of detail about these. I would kind of just have a general idea of what they do.
Also, kinesin 14s, they are mindset.
Yeah. They are not on this diagram, but they are on that first slide. So when we send this out, if you want to write that down. Okay. So how does kinesin 5, the question, which is also like egg 5 or includes egg 5, push spindle poles apart? Yeah. So I'm gonna draw this. So if this is our cell, right? We have our centrosome here and here. We have our interpolar.
Definitely not drop the scale.
Yeah, it is. So this is our cell. Gorgeous. These are our centrosomes. We have interpolar microtubules, and then egg 5 binds on them. And what they do, this is the plus end of our microtubule. Both of these want to walk towards our plus ends, but we can't elongate this middle section, this length of the term. So when this walks this way, this walks this way, the effect that has is these have to get further apart. So egg 5 would sit essentially at some point on the plus end of both of our ends. And that push our points further. Does that make sense?
So this is an exam question. Yeah. Yeah. Exactly. Basically, what she's looking for is kinesin 5, egg 5 in this case. They have 2 sets of motor domains on either end of them, which is what allows them to bind 2 microtubules simultaneously, which is then how they're able to walk towards 2 different plus ends, which is then how they move microtubules with respect to that. So if they walk all the way to the plus ends, they want to just sit like this, and the only way that they will is if these interval microtubules are pushed as far away from the so as it walks. When I do these on exams, I would always think, well, like my fingertips to the front of my heel, when we're walking to the plus end, right? The only way that we'll get closer to the plus end is if we move our spindle pole part of this. That's how I thought of it. That's helpful. Work for somebody in my office hours. Okay. Yeah, so we just covered that, but that's what that is. Oh, yeah. What happens if we have a loss of function in egg 5? I can see why you would think that, but did you have a different guess? Right. So we have opposing forces happening with our motors in the cell. And they kind of create a balance of like inward pushing force and outward pulling force. And our egg 5 is the thing that's pushing our spindles further apart. If we lose that and we don't have that outward pushing force, it's overcome by the motors that are pushing in. So our spindles will essentially just collapse onto each other and our cell will die.
And that, so this is our normal cell where we have that equilibrium of forces that's maintaining its shape. And then when we don't have our egg 5, if we get rid of it or have a loss of function, these are our spindle poles right here. And these are all of our microtubules in our DNA. They just completely collapse onto each other. But
So there's always like an inward and outward force.
Yep. yeah, because we have different motors that are walking in different directions, some that are pushing, some that are pulling. So we want to be more or less in equilibrium, or if we're not in equilibrium, like just not enough that we can like push apart a little bit or pull in a little bit, not a total loss of one of those forces. Okay.
Alright.
Does anybody, sorry, before, just does anybody have any questions on that? Okay, and this isn't in the slides, but I am gonna go over it really quick. Because this was a problem set question that I had a lot of questions about at my office hours. So, dynein is another motor that is minus end directed, that sits on the outer cortex of the cell. So, like, or just drawn over there. Dynein sits somewhere on the outer cortex. It's assigned to one of our like roles, and it wants long towards the minus end here. So, but it's found right here. It is stuck on the cortex. So the analogy that's something about like really like is you can picture that as like a person standing on a dock, and a person is here in the boat. This is a boat. So our dynein is not moving down. It's pulling the microtubule, kind of like a boat, as it's walking towards the mindset. And the effect of that as is pulling this out. Does that make sense? I think so. Huh? Yeah, so. Okay. Right. This is our dock right here. This is our boat in the water. And instead of our dynein walking along and moving this, it is pulling the microtubule like a boat as it's walking towards the mindset. And as it pulls that boat and walks on the microtubule, it's pulling the central closer. Does that make sense? Because it has to stay where it has to be anchored on it. So I just like that one problem. Well, what that helps you with
Basically, you're very close. But that right here, essentially happening is the section is going back. So we have other mechanisms that's happening here. This other. Essentially what's happening is the fact that this is originally and it's connected to all of these other these two main pulling force towards itself, and so if you can send anything, piece of you don't want to have anything in the middle of the cell in order for the cell to split. But hence, because if it's in the middle, it's just like a micro section. You could say, and we want it as far as the whole that side of the cell as possible, so that you can have. It won't be 100%. It won't be perfect like that, but you're looking for at least all of your people.
All right, so today's review will only go over first stage of cell cycle. And then Antonio's and Gabe's on Sunday will go more in depth on the second part. So cell cycle overview. So you go from G1 to S to G2 to M, or to G0. And so G1 is start of the cell cycle. It is the growth phase. So the cell is preparing for DNA replication. It's checking for enough nutrients, enough cell machinery to see if the DNA has no mutations, to see if everything is functional and optimal for the division to occur. Synthesis is when the cell is replicating its DNA. It is to make sure that you have copies of your in eukaryotes chromosomes to both daughter cells. And so there is a very important end of S phase checkpoint. And then once that occurs, you move into G2. And G2 is a second growth stage right before M phase. So the cell is preparing for segregation of chromosomes and also has a G2M checkpoint. And so once we move along to the M phase, you have 5 different stages or 6, depending on the source you look at. Prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. And cytokinesis is the division of 2 new daughter cells. And also you have G0, which is non-dividing state, which the cell determines that it's not yet fit for the replication process to occur. All right. So an important factor of the first stage cell cycle or cell cycle in general, CDK. Laura likes calling them the master cell cycle regulators. And different CDKs push the cell through the cell cycle when active. There are specific ways that CDKs can be regulated. We talk about different ones in class. Right now, we're going to be focusing on cycling levels, phosphorylation or dephosphorylation, which doesn't mean activation or deactivation. And yeah, and also CDK inhibitors, but you need activating phosphorylation and cyclin for CDK to be fully active, and CDK will always function in a CDK cyclin complex, right? Never mind. And so, yeah, right. Yeah. But and so checkpoints exist in order for the cell to check for different things, but how they happen is there's a halt of CDK activity, which then doesn't allow for cell division processes to occur. And so CDKs are able to target different substrates, and the different cyclins that are expressed help CDK focus on the correct proteins they need to reach. Any questions? Any comments? Okay. So cyclins, this is a really good chart. If you didn't have it on, or actually, was this on exam 2? You don't need to know when cyclin release happens or whatever, but it's important to understand the levels of them and how each different cyclin affects each different stage of the cell cycle. Like I said, cyclins are very important. Not only do they bind to CDK and help activate it, they help CDK know which proteins to target in order for the different processes to occur. And so during M phase, if CDK needs cyclin B to phosphorylate something or to activate something, then cyclin B will be expressed in a higher amount. And so cyclin can only be controlled at transcription levels and ubiquitin mediated proteolysis, while CDK is more broadly regulated. And so here's the complex that we were talking about. CDK alone is tucked, or I mean, its activation loop is tucked inside of CDK, and it cannot bind to anything. Once CDK becomes bound to cyclin, then it undergoes a conformational change, which exposes the activation loop. Laura really likes going back to this concept of structure leads to function. If there's a change in structure on the protein, then either it misfolded and it won't be able to perform its activity, or it changed for a reason and its activity is modified. And so CDK can only phosphorylate other proteins when bound to cyclin, and it's not yet at this stage that it's officially active. CDK activating kinase CAC is what phosphorylates CDK's activation loop, and then that allows it to be active. Can I help you? No, I'm kidding. And so CDKIs can block CDK from phosphorylating target proteins or from CDK binding to substrate. And that is another way of CDK activation limitation. All right, so cell cycle arrest. If you find DNA mutation G1, what happens? P53 gets activated. P53 is a very important protein. It is a transcription factor and it's tumor suppressor and usually responds to DNA damage. P53 upregulates transcription of P21. That's probably the most essential thing out of P53 that you need to know. Because P21 is a molecule that is able to inhibit CDK, not molecule protein, sorry. And so if DNA damage is deemed irreversible by P53, or by the cell, P53 signals for apoptosis, and then damaged DNA transcription doesn't occur. And P53 is constantly being degraded if inactive. And so there's a little pathway that shows how inactivation of CDK cyclin occurs through P53 and P21. But yeah. So 2 questions. Why is P53 a tumor suppressor? What you got? What's P21 do? I just want to know. Can you? And P53 is widely studied medically due to its function in tumor suppression, and modified or mutated P53 proteins can then lead to overproliferative or non-checking cancerogenous phenotypes, which is why it is widely studied. And so what would happen if P53 was no longer available? Regardless of DNA damage, right? DNA damage, which is essentially what P53 is leading or doing. It's checking for that damage. And that's why people dump money into P53 research. All right. So G1 phase. So the cell is checking to actively see if it has enough nutrients, if it's big enough, if it has enough cellular machinery, proteins, enzymes, whatever you name it. And if its DNA is not damaged. In early G1 CDK complexes form, but they will not be active just yet. And that just means G1 cyclin bound to CDK. And so before that complex is active, pre-RCs form. During mid to late G1 restriction point, that's after CDK becomes active, and then the cell must pass such restriction point in order to commit to the cell cycle. It either commits to it and goes through with whatever it has, or it goes into apoptosis if it deems that it is not going to make it. Or it can also go onto a replicative state, but if it stalls for too long for whatever reason, doesn't have enough nutrients, doesn't have enough resources, then it eventually also undergoes apoptosis. And then in late G1, pre-IC forms when CDK activity is high. So pre-RCs, they form during early G1, like we just said. And then the first protein, the vital protein here is ORC, origin recognition complex. And it will bind to the origin of whatever sequence of DNA it is looking at. Once bound, ORC is going to recruit CDC6. And then the ORC CDC6 complex will then recruit CDT1. And then it'll recruit MCM. MCM is the helicase for this complex. And so multiple MCMs can be recruited to unwind DNA in both directions. Oh, okay, going back to this. This doesn't happen in only one place, like we saw in different problem sets. There are multiple different origins of replication that fire at different times. And for that to happen, you need multiple pre-RCs to be formed. Okay, restriction point, it is the point of no return. And the cell will then commit to cell cycle. Everything's already checked for. You have enough nutrients, you have enough machinery, enough proteins, enzymes. G1 cycling CDKs become active, and RB, which is that big molecule holding on to E2F, is phosphorylated. Once that happens, there is a conformational shift, and RB is forced to let go of E2F. E2F is a transcription factor that upregulates proteins needed for S phase, which is when replication happens. And it is kept sequestered by RB until there's enough G1 CDK cyclin or CDK cyclin complex in the cell. And after that, the cell then makes the decision of whether continuing with the cell cycle or not. But E2F is very important because it is a transcription factor that transcribes essential proteins for S phase. And so there's a little pathway over there, which is basically just the image version of everything I just said. And phosphorylated RB becomes, or its function is inactivated. Its original function being holding on to E2F. And so pre-IC formation, once G1 CDK becomes active, more proteins bind at the pre-RC. So for a pre-IC to be formed, you need a pre-existing pre-RC. CDK is required to get these proteins to bind. And this is where the, that's basically the taking form of the different cyclins being expressed. That's just an example. G1 cyclin is being expressed at a higher level right now, which is then what leads to G1 cyclin CDK complex being formed, which is then what leads to the pre-IC formation to start. And these complexes are larger than the pre-RC, obviously, because they're being built upon them. And so they are called the pre-initiation complexes. And so once the cell is ready for replication to start at this specific origin, a polymerase will get loaded and DNA replication will start. And this beginning of DNA replication instates the S phase. And so you are past the restriction point and you are committing to it. Okay. And yeah, just a little side note at the bottom. Firing is a common term used for when replication starts at an origin. And because of the fact you have different origins, you can have them start different times. They're not all going to be timed at the same time. And this is very important because as we went over in class, 3 billion base pairs, that is a lot of DNA to be replicated. And so you just need it to be a quick process in order for your cells to remain healthy and divide.
Would you be able to go back? The woman said. Yeah, the woman.
All right, so we have some true or falses and one normal question. So in response to DNA damage, cells should increase concentration of CDK inhibitors. Why? All right, number 2, in response to DNA damage, E2F is active. False. Why? Or leads to transcription. All right. Could E2F be a cause of cancer, and can a cell divide if E2F does not function? And if your cell doesn't have enough, or if your cell has enough nutrients and energy but doesn't have all the machinery it needs, what happens? No, post D2F activation. Good job, Matri. All right, fourth question, true or false? PRCs can form at the end of G1 after CDK is fully activated. Why? And then what happens once they're already formed? Because of the CDK activation. All right, so that's about it. We're not gonna bore you with those questions. You can obviously ask questions if you have them. But thanks for coming and yeah.